Hello, good afternoon. I'm Carrie Moss Stearns. Welcome to this news briefing from the 250th National Meeting and Exposition of the American Chemical Society in Boston. We are joined today by Dr. Robert Grass from ETH Zurich, who's going to talk to us about how to preserve digital information with DNA. Dr. Grass?
Thank you very much for the kind introduction, ladies and gentlemen. We all store our information, our work information, our personal information on our computers. Most of us don't have photographs or don't have real photographs of the pictures we take. We don't have paper copies of the work we perform. Everything is online and digital. And we somehow know that this information is very fragile, that it disappears in time suddenly. Everybody certainly had an incident where he lost part of his information or large parts of his information. And so we have to think about how do we preserve information for a longer time. If we go back to the past, perhaps 500 years, there were people, monks usually, who really tried to preserve information with the idea of preserving it for future generations. Of course, here we're not talking about what kind of information we have to preserve, but how we shall preserve information. We know that paper is stable for around 500 years. The oldest books we know are close to 1,000 years old. But we have to think in the, let's say, enormous amounts of modern information, what are the ways that we can preserve them in a really stable way? There are a few ideas into this, and there have been a few ideas into this. I think the most logical way is to see what's the oldest information we have, we can find. How was that stored? And then you go to, let's say, Greek cultures, you see inscriptions in stones, which is very stable. If you then think away from, let's say, man-made information into really storage of information, you find fossils. Fossils contain genomic information of past species and very large amounts of information of past species stored in DNA. Most of us have probably seen Jurassic Park, where it's not what's done in Jurassic Park can't be done. DNA is not stable in amber fossils and is also not stable for 100 millions of years. But there are fossils 100,000 years old or 700,000 years old, which have information in them stored in DNA. And so what we do, is we take this idea of storing information in DNA. We know that it's extremely stable if it's stored correctly over hundreds of thousands of years. And we then have to think, well, how do we not store genomic information, but digital information, photographs, our Word files, our documents, the academic work we do. That's what and so we have to figure out how we can get this kind of information stored in a way which is as stable as the genomic information found in those fossils. And so we both looked at the translation of the digital information into codes, which then translated to a sequence of DNA base pairs, where more or less you go from translating something which is a sequence of zeros and ones to translating it to a sequence of A, T, C, and Gs in the structure of the nucleic acid. So one part is really this translation. And the second part of our work relates to how can we really physically store the DNA molecule so that it's as stable as it's found in the fossils. Because if you have DNA, if you work with DNA in the lab, have it in the fridge, in the fridge it's perhaps stable for a year, perhaps for 2 or 3 years. If you have it outside of the fridge, it's relatively fragile over the course of a few weeks. And that doesn't relate to the high stability DNA has in fossils, 100,000 years. So what's different, we had to sort out what's different between these two storage media. And taking the idea of the fossil, generated an artificial fossil where the DNA is encapsulated in an inorganic material where it's protected from water, oxygen, and it doesn't degrade as fast as it does in solution. That, by the way, is an image of our inorganic material in which we have encapsulated DNA. The material we use is silica, glass, in the end. The reason for choosing glass is that it can be very easily synthesized in water at room temperature, and still it's an extremely inert material. I mean, our houses are protected from the environment by glass. It's extremely resilient to the environment. And here we do the same. We use glass, small glass particles, they're only a few nanometers large, in which we chemically entrap DNA molecules. And in that entrapped state, they're almost as stable as they are when they're entrapped in fossil bones of a few 100,000-year-old fossils, which are known to be stable over that time frame.
Thank you, Dr. Grass. Do we have any questions from the audience?
So it's Kath O'Driscoll from Chemistry and Industry magazine. Just to ask, I guess the big problem with using DNA storage is that it's so expensive. Is there any way, is there going to be any way in future of actually reducing the cost and the labor of actually doing that?
For sure. The question is, there has to be the need of doing it. Because intrinsically, the cost of the DNA synthesis, of the consumables you need, of the chemicals you need, is not extremely expensive. It's at the moment, there are only a few companies worldwide which really do DNA synthesis to an extent where you can write a few megabytes of information. And there's no market or movement. It's the hard disk industry has done 50 years of development in reducing prices, in being more efficient, in making the materials, in selling them. It has a big market. And DNA synthesis, the market of that is really small. We see a very big development on the side of writing from DNA because of all of the biomedical research related to understanding the structure, the functions of DNA. All of the sequencing technologies are moving very, very rapidly. And something very similar would be possible on the synthesis side, but there has to be, let's say, the real need from a political or public or user or an academic field to push that in that direction. But it's certainly one of the biggest constraints in this field, as in most academic fields, by the way. The scale of getting it large enough and cheap enough so that it can really be integrated into everyday product. And I think the way to solve that, or what we are trying to solve, is not by starting with the dream of wanting to store gigabytes of information, but more thinking about what information do we really need? Do we need gigabytes of it? So we can start relatively small. We start even with barcodes, which we store. That's not in the, let's say, storage of information, but a barcode is also a kind of information. And we can generate with these particles barcodes, which are very stable in the environment. And we can use them as tracers for products, for environmental processes, for chemicals. That's a different motivation behind it, but let's say from the technological side of it, it's what will have a much faster transition, or let's say an early transition, and then we think that we can grow with the need of that and lowering the costs of the DNA synthesis for that. We believe that we can grow in that direction.
Any other questions? And please state your name and affiliation.
Bela Buslig, ACS News Service. Now, I'm going to go out on a limb on this one, but it needs an answer. When you're storing data, any kind of data, particularly DNA, it's stable. But the problem is retrieving that data. And when you're doing DNA and you're trying to retrieve DNA data, there's the first thing that can happen, duplicating it any which way, enzymically otherwise, is there are mutations. And then the mutations will give you something a little different, so it can propagate. How many? I mean,
okay.
Just
I think I can.
I need. How many redundant copies would you like to save, or do you have to save to get the original information? And will it, in 100,000 years, will that information make any difference to anybody?
Yes, I think the two answers or two parts to that question. One is a very technological one, and I was in a lucky position when I started with this research, that one of my friends, he did a Ph.D. in electrical engineering in communications theory, and there you were always thinking about mutations. I mean, our hard disk, every millisecond it makes errors, and you have to have ways to anticipating those errors and then introducing redundancy to remove those errors. And the redundancy added is done in a very smart way, that you don't add redundancy by copying the information, but there are mathematical codes where you can add a little redundancy and you can still correct any error in any position of your data. So we implemented that into our storage technology. So we had a look at, so we wrote the DNA, encapsulated it, and heat treated it to simulate damage. And then reread it and had a look if the codes we implemented to, let's say, the predicted error we assumed, if we could really remove that after rereading the information. And we could fully recover the information after about the decay by 4 half-lives of the DNA. In other words, about every 50th DNA base pair was wrong. And by this implementation of this, what's called forward error correction, we could correct for that error in the decoding stage. So that's implemented. And so we are not, we can work with a certain extent of mutations. Not endless mutations, but let's say anticipated amount of errors or mutations which happen either during writing, the storage, or at the end of the reading also errors happen. And I think the second part of the question that I somehow heard is, you didn't state it exactly, but it's a very common question. If we think in 10,000 years, or even 100 years, perhaps people find the DNA, but they don't know how to get from the DNA back to the digital information, to the image file. So how do you do that? How do you guarantee that you have a method which can be decoded at future time frames? And in the case of DNA, there are two answers to that. One is we assume, perhaps stupidly, but we still assume that future generations will always be interested in being able to read DNA. Because the second thing, you have to have an instruction how you get from the DNA back to the, and that's a problem common with all storage formats. We all know our VHS cassettes where there's no player and you can't do, I mean, the information is there, but you can't read it. And so you have to give instructions on how it's read. And every information storage is valuable if the amount of information you can store is much larger than the amount of instructions you have to give to read it. Because then you can give the instructions on a different, also stable storage media. There are many. There's inscription in stones, which is extremely stable. The density you can get into it is really small. But you can, let's say, give instructions on how to resolve the method which stores the larger amount of data. I think a very nice example of that is the Rosetta Stone, which was found in the Napoleon Wars, where before it was impossible to understand the Greek language or the drawings. and with the stone, which had a copy in ancient Greek and in 3 languages, if I remember correctly.
Familiar with it.
Suddenly, it was clear. So we need different levels. The DNA alone is not the full solution.
Basically, I agree with you fully there. But well, you brought up the Rosetta Stone. It was a very, very small amount of information, small text, and it was relatively simple. But imagining something like the Encyclopedia Britannica. Now, when you read the Encyclopedia Britannica, you don't always want to read the whole thing. You don't want to retrieve everything from it. So when you've got smaller pieces of information, it's actually easier to handle than when you have to copy the same thing over and over. I mean, look at the Bible, how many copies there are. And there are 2 that are the same.
Yes, indeed.
Well, short of printed stuff. But
of course. I think one of the next things we are doing is thinking one side about the instructions, but I think that's, as chemists, that's difficult. I mean, it's not a chemical problem in the end. But to think about a file system, or how do you assign certain bunches of information to certain DNA strands, and how, because the advantage of all of our main storage devices is that information is really stored in a sequence, physically in a sequence. So I can read from here to here, and then I have my information. With DNA, that's different. I have a liquid, and at the moment, to get the information back, we have to read all of the information that's in the liquid. The solution is, okay, I can put a second volume of liquid, but then I still have to know which ones where. And one of the advantages of DNA storage is that the density you can get is very high. But then if you're going and assigning parts of DNA to certain liquids, you lose that advantage So what we're doing is, there are methods, we are, let's say, perfecting them at the moment, to select certain parts of information from a bunch, from a solution. One is by magnetic selection of certain sequences. So let's say you make a file system where you have encoding particles which select for a certain file, and you then only read that file that you have selected. And so those are, besides, of course, there's a really a cost issue in getting this into a fully scalable. Some of the questions, more technical and theoretical questions, is exactly how you can solve these issues you brought up.
Well, thank you very much.
Pleasure.
Hi, Josh Howjago from New Scientist. So when you began the introduction, you were talking about how DNA is one of the oldest sort of places that we can find information. But I was just wondering, I mean, that seems to me to be sort of, in some ways, a sort of a funny way of thinking about it, because that's just almost, it could be thought of as just a sort of an evolutionary fluke in some ways. And I was wondering, if you could you not just sort of design a molecule de novo? Why assume that DNA is the best?
I think that it's wrong to assume that DNA is the best. And there are people who do that, who do de novo. There's in polymer chemistry, there's a field that's growing there in really, let's say, block copolymers where you really design every individual step. It's very difficult. At the moment, the big advantage DNA has and its history has, we know that it's stable. With all other molecules, I mean, you can test them with modern tests. Like we do the same with our experiments, we do accelerated aging to get some proof that it's really stable. But accelerated aging has assumptions behind it. And if you want to be really sure, and we have a molecule for which we really have real data for thousands of years, and people have really used fossils from 8,000 years to calculate the kinetics and the decay of the DNA. We have that. There's no assumption behind knowing that it's stable. And that's the largest advantage it has using it. Besides the possibility that the reading and writing, especially the reading side, is really established by our current... These things wouldn't be possible if we wouldn't have the, let's say, big urge into human genome sequencing. That's really a requirement that works, that we can have these tools available to do that. In chemistry, people do that, but in terms of information you can read, and let's say the selectivity, the variability you get, one example is protein analysis. Protein analysis can be done by, let's say, mass spectroscopy methods, but it's much, much more difficult. The puzzles you get from your mass spectra to getting back to the original protein structure is much more difficult than in DNA, where you kind of have a sequential reading during the sequencing step. And also at the moment with Oxford Nanopore, this is advancing further. It's getting faster, longer, more accessible for smaller labs, perhaps even accessible to an individual user. And I think that there's the other technologies also have their right place, and it might be that you can make much more stable, because I think your thought comes from the direction that DNA in our life doesn't have to be extremely stable. It's really by coincidence somehow, if it's encapsulated in calcium phosphate, it is a stable molecule. It wasn't this. It has to have some stability, but not in that extreme sense.
Thank you for your questions. This archive version of this session will be posted at bit.ly/ACS Live Boston. Please join us for our next press conference today at 3 p.m. on a new way to help detect traumatic brain injuries in soldiers and athletes. Thank you.